and very carefully interpreted. And the other thing I've always enjoyed about her work is that she is very diverse in what she approaches. So while the bulk of her work probably could be put under the rubric of HIV and drug abuse, she's used that platform to look at a lot of other processes that involved many different neurodegenerative diseases. For instance, she did a lot of work on the cysteine histidine-rich protein known as NIMH. When she started working on it, there weren't that many people interested in it. And then after some period of time, there was a big explosion in the field in multiple different neurodegenerative diseases where pinch became important. So she was sort of really early on involved in that. She's also been involved in some traumatic brain injury work using accelerometers with a football team there at Temple. To really do some elegant work tracking brain concussive types injuries and subconcussive types injuries and the long-term outcomes or longer-term outcomes associated with those. Recently, she's had some really interesting work at looking at sumodylation involvement in the innate immune system, which is a very interesting post-translational modification that really hasn't been looked at as a regulator of innate immunity as of yet, but she is going there. And of course, today she's going to talk to us about astrocytes that she's always had a large interest in, and especially in the roles of astrocytes in regulating brain damage and in regulating brain energetics. So the title of her talk today is The Self-Serving Role of Astrocytes. It's all about me. So, thank you, Dianne.
Thank you, Norm. You made me sound really good. Wow. So it's really good to see everybody on today, even if most people are named Heather Thomas. So what I want to talk to you about today is it's a lot of in vitro work. So there's not going to be any fancy animal work that I'm going to talk about today, even though we are doing some animal work. There's not going to be a lot of work on HIV participants or cocaine users, but this is just some really cool to me metabolic lipid dysfunction studies that I'm really enjoying. And the reason I titled my talk, The Self-Serving Side of Astrocytes, It's All About Me, is because during the course of a program project that I worked on with Dr. Khalili and others, and Norm included, we discovered some stuff about astrocytes that I was not aware of. So when astrocytes are challenged with things like HIV, cocaine, anything that induces inflammatory response, reactive oxygen species, they kind of say, okay, fine. And they upregulate all of their metabolic processes and really ramp up. In doing so, what happens is they move away from their neurotropic phenotype and they stop making things for neurons that neurons rely on, like lactate and cholesterol. So today I'll be talking about how HIV and cocaine converge and disrupt both astrocyte and neuronal lipid metabolism. And just a quick search, and I know many of you are familiar with at least some of these articles. There's over 3,500 articles about brain HIV and cocaine in the literature. So we all know that metabolic disturbances in the brains of people with HIV are common. We also know that people who use cocaine have different types of metabolic disturbances. So when you put them together, you have effects on virtually every cell type in the brain. You have chronic neuroinflammation, disruption of neurotransmitter signaling. Microglia can be infected, whether it's a latent infection or replication happening then. And also cocaine can induce viral replication in different types of microglial signaling. Blood-brain barrier is impacted, and of course astrocytes are implicated in low level, not so much productive HIV replication, but they are also a real workhorse in the brain to do much more than support neurons when you get down to the signaling level. So as I mentioned, astrocytes, let me just move this over here. Astrocytes supply key metabolites to neurons because neurons have a really low antioxidant capacity compared to astrocytes. So neurons can handle some excitotoxicity, they can handle some challenge, but their capacity to do so is limited compared to astrocytes. Like I said, astrocytes are usually like, bring it on, we can do this. So astrocytes make lactate for neurons. Astrocytes also, see these. Astrocytes also make a lot of the cholesterol that neurons need, and neurons rely on shuttling of cholesterol through the ABCA1 transporter in astrocytes hooked to ApoE to take cholesterol up and use it for different processes. And when you have a depletion or deficiency in the astrocyte-derived cholesterol or lactate, neurons have a problem. So in my laboratory and in collaboration with some other groups that I already mentioned, we have at least 3 different ways that we've approached this. One story that I'm going to talk to you about today is that when neurons are challenged with HIV or cocaine, you see an increase in reactive oxygen species, you see autophagy increase, and you also see fitness decrease in the neurons. They increase their LDL receptor to try to pull in more cholesterol, and they also produce and accumulate a lot of peroxidated fatty acids that once they've quenched their capacity to detox, they just spit them out. Now, and this is when neurons see HIV and cocaine. Now, when astrocytes see HIV and cocaine, what they do is switch from glucose metabolism to beta oxidation. They increase their ATP, they decrease making cholesterol, they decrease lactate, they decrease ApoE production and ABCA. So they keep everything they're making. They don't give anything to neurons anymore. Now, what I want to talk to you especially about today is neuron astrocyte shuttling of peroxidated fatty acids from neurons to astrocytes, and how important the citrate transporter in astrocytes is for this to be a healthy process for the astrocytes. So I know the resolution of this figure is really bad. I tried to fix it, but I did not. I was not successful. So Bianca Cotto, who was a graduate student in my lab years ago, she's now a postdoc at Rockefeller. She published a paper showing that lactate generated by astrocytes was reduced in response to exposure of astrocytes to cocaine or to Tat. She also showed that astrocytes decrease their ApoE and ABC1 through an LXR nuclear receptor mediated function that is also responsible for sterol regulatory binding protein. And this is going to be important down the road in my talk, that is responsible for transcription of a citrate transporter called SLC25A1. So I know I'm throwing a lot of words and stuff at you, but it'll make sense, I promise. Okay, so what I've told you so far is astrocytes shift from producing lactate and cholesterol to making acetyl-CoA for increased ATP production. They decrease their ApoE and ABCA1 and cholesterol, and the increased acetyl-CoA that they make is converted to citrate that can go into the citric acid cycle to increase this ATP production. So basically what happens is when astrocytes are challenged, they increase their metabolism and change bioenergetics. Neurons induce autophagy, reduce spare capacity, and quickly quench their antioxidant capacity. Neurons also generate excess toxic peroxidated fatty acids and expel them in the form of lipid droplets. Astrocytes take them up and use the lipid droplets for processing as an energy source. This is just to remind you of what we're talking about here. So this is the cytoplasm, and this is the mitochondria, and this is an astrocyte. So usually glucose goes, there's a lot of steps in between pyruvate down to acetyl-CoA. Citrate comes in from the mitochondria and works with acetyl-CoA for fatty acid biosynthesis. In the mitochondria, on the other hand, citrate can be shuttled back from the cytosol into the mitochondria, and through an interaction dependent upon IDH, which is mostly known about in glioblastoma, the citrate's converted to isocitrate, blah, 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 blah. You go back around and you have the proper running of the TCA cycle for energy production. Now, this back and forth shuttle here is what we discovered is super important for astrocytes to be able to do their job and take care of themselves, even at the expense of neurons. So this is kind of like the graphic abstract, if you will. So what I'm going to talk about to you today is how citrate, when you mess it up, this doesn't work, and the astrocytes are not able to process the excess peroxidated fatty acids from neurons. Before I get into the experiments we conducted, I want to just give you a little bit of experimental design because I think it'll help you visualize what we really did. So the cell sources we had were human fetal astrocytes and neurons, and this was way back when you could actually use these. Now we can use them again. We also used iPSC-derived astrocytes and neurons, and in some of the experiments, we tried it in SHSY or N2A cells, differentiated into neuronal-like cells first to see if it would work. Now, in some experiments, we exposed neurons to Tat, to cocaine, to both coke and Tat, to NMDA as a positive control, and alpha tocopherol to block the fatty acid oxidative toxicity. We also exposed neurons to all of these components that I said and collected the media and called it neuron condition media. We took this media from all the different types of treatments and exposed astrocytes to it. In control experiments, we also exposed astrocytes directly to cocaine, Tat, both NMDA, alpha tocopherol, to make sure that it was what we were seeing directly from neurons to astrocytes and not what astrocytes were doing because of the cocaine or the Tat. And we used transwell systems that we'll get into a little bit later. So I just wanted to give you an idea of what we did before I jump into the data. The first series of slides we're going to look at are neurons treated with cocaine or Tat, and we measured mitochondrial reactive oxygen species. And just a reminder, alpha tocopherol is a lipid-soluble antioxidant that blocks lipid peroxidation. So what we saw here is that this is control down here. And so in green, you see row 1, 2, 3 for mitochondria, mitosocs for mitochondrial reactive oxygen species. And what you see is that in the control, you don't have a lot going on. However, when you treat the neurons with Tat, coke, both, or NMDA, you have a dramatic increase in mitochondrial reactive oxygen species that alpha tocopherol wipes out, brings it back down to normal levels. Neurons also produce a lot of fatty acid, peroxidated fatty acids in response to coke and Tat that is completely blocked by alpha tocopherol. And you can see it here. And I want to just point one thing out to you, and you guys can help me keep track of this as we move through. Red is Tat, blue is cocaine, green is both together, and purplish pink is NMDA. Now, I'm surprised about this. Everybody's surprised about this. We don't see any additive or synergistic effects of coke and Tat ever, ever, ever. But what we do see is that cocaine tends to be the driver in almost all of our experiments. And it follows pretty tightly with NMDA. So this is not a Tat specific thing going on. Cocaine seems to be not in all cases significantly the driver, but the driver. So we must jump to a model of HIV and not just one HIV protein, which we're doing now. We're using the Eco HIV mouse model with Coke. So we'll have more data soon. But this is still pretty cool, I think. So not only do neurons have stressed mitochondria, they accumulate fatty acid, peroxidated fatty acids, as you can see here. And once again, you see Tat driving it. But this accumulation within the neuronal cells of peroxidated fatty acids induces autophagy. So these are autophagy markers here. You can see LC3B1 and 2, phosphoAMP kinase, with and without alpha tocopherol. And you can see here that in the presence of cocaine and Tat, neurons do induce autophagy. But the cool thing to me is what they do with all of the accumulated peroxidated fatty acids that they produce. They spit them out into the media. And as you can see in the bottom three, G, H, and I, you can see that what I've shown you here is the blue is control. This is a flow experiment, and this is Tat treated, Coke treated, and both. You can see an increase in the number of BODIPI labeled particles released into the media. So what we did is we labeled these fatty acids in the neurons. They spit them out, and then we put them on astrocytes, and we see in the next slide that astrocytes take them up. So here you can see that control has very little neuron-derived fatty acids that astrocytes have taken up. So we're looking at a picture of astrocytes here that have been treated with neuron-conditioned media from neurons that were exposed to Tat, cocaine, both, and NMDA, that had label incorporated into their newly produced peroxidated fatty acids. So as you can see, without alpha tocopherol, our antioxidant, you see that astrocytes take up a lot of the neuron-derived in red fatty acids here. And once again, here's Tat, here's cocaine. And while these are both very significant, you see cocaine just tends to pull out in front just a little bit. This is one of my favorite pictures. I just love it. Okay, so the next thing we wanted to do is look at what astrocytes do with all these fatty acids. Like, what happens to their metabolism after they're taking them all up? So we did some simple oxygen consumption response experiments using the seahorse. And just to explain this to those who don't use it every day, what you do is you take your cells and you treat them with different factors that will show the response to various points in the mitochondrial respiratory cycle. So you treat it with oligomycin, which inhibits basal oxygen consumption rate, FCCP, which is an uncoupling agent that gives you the value of the maximum oxygen consumption rate. So here's A, B, and C, and finally in C, rotenone, which is a mitochondrial complex 1 inhibitor. Now, in these experiments, and this is just to show you how we did it, we used palmitate as a fatty acid substrate for fatty acid oxidation, BSA as a non-fatty acid control, and we used ETXR as a fatty acid oxidation inhibitor to show what's going to happen. So what we're showing you here is in astrocytes that weren't exposed to media from cocaine or Tat-treated neurons, they had a very normal response in all cases. When you give Tat, what you see, increased fatty acid oxidation here, same for cocaine, same for Tat and cocaine. Now let's look at the interesting data. So the way I've broken this down is to control, meaning the astrocytes were treated with control neuronal media, normal neuronal media, Tat, cocaine, Tat and cocaine. Basal oxygen consumption rate went up for every condition, Tat, Coke, Tat, Coke. Same for maximal oxygen consumption rate, ATP coupled respiration went up, but non-mitochondrial respiration did not change. So once again, as you see, Tat, Coke together all the way across the board pretty much did the same thing. Illustrating once again that we've got to really drill down on what cocaine is doing in the context of HIV itself, not just one viral protein. So now I'm going to move into the newer data that Dr. Srinivasan and others in my lab and Dr. Garcia have been working on for quite a while now to look at least one mechanism or one process that must occur for the astrocytes to be able to take up all of this junk from the neurons and make it work for them. And what we're looking at now is the SLC25A1. Now that's a citrate transporter. So citrate that comes in from the pyruvate cycle and goes into acetyl-CoA. What can happen is if the mitochondria need more citrate, this transporter can pump it in from the cytosol and vice versa. If the cytosol needs more citrate to actually go back and make more pyruvate for lactate or cholesterol, citrate, if there is enough in the mitochondria, can be pumped out into the cytosol. Mostly it goes this way, but it can go the other way as well. So why did we pick SLC25A1 as one of our potential factors of regulation for all of this? Previous studies have shown that disruptions in this citrate transporter have been linked to at least 11 different CNS diseases characterized by lipid utilization disruption and low energy production by mitochondria. We know that citrate is really important in both components, cytosol and mitochondria. It's also important in the nucleus, but we're not going there today because I don't really understand it yet, and we don't have any data. And we also know that cytosolic citrate is the precursor for acetyl-CoA that leads to cholesterol and lactate production that neurons need. So if you don't have enough citrate in the cytosol, the neurons are going to be out of luck. And what we've shown in our data, this is the take-home message. I'll show you the data. Loss of citrate transport during increased peroxidated fatty acid uptake impacts the astrocytes' ability to process the fatty acids and provide neuronal support. So what we did first of all is just knocking down this transporter. And what I want to point out to you that was really interesting to me is when we took astrocytes and knocked down this transporter, well, first of all, we showed that we knocked it down. But when we took astrocytes and treated them with neuron conditioned media from Tat neurons or cocaine neurons, the astrocytes in the normal condition increased this transporter. So it showed us and confirmed that this transporter is super important for astrocytes to be able to handle all of these peroxidated fatty acids. So when we treat astrocytes with a neuron media that has all the fatty acids in it, they increase this transporter. Now, what we want to know is which way is it pumping? I don't know. We're working on that right now. So this was a really cool result. What we also see is that when you knock down the citrate transporter in the astrocytes that have been fed with these neuron-derived fatty acids, if they don't have that transporter, they just accumulate these fatty acids and they fill up with lipids. And this is not a good thing. Now, what we know is that in normal aging, in Alzheimer's disease, in Parkinson's disease, in ALS, astrocyte and microglial accumulation of lipid droplets is a normal thing. So what we've shown here is that if you can't make citrate go where it's supposed to go, you have enhanced increase of this lipid droplet accumulation in astrocytes. So even though you see it in normal aging, it's exacerbated by things like we predict is exacerbated by things like HIV, cocaine use, abnormal aging associated with HIV infection, which is a new direction that we're looking at right now with others in our department. So then we wanted to say, okay, we know what astrocytes do when they have to process tons of peroxidated fatty acids from neurons that were exposed to bad stuff. What happens to their bioenergetics? And what we found is that if astrocytes can't transport citrate, their intracellular cholesterol goes down. Their as you can see here, intracellular cholesterol goes down, suggesting that they're not making any more. Their extracellular cholesterol goes down, and their ApoE goes down. So this is to show you that if the astrocytes can't transport citrate properly, not only does it decrease anyway, it decreases even more in the neurotropic capacity. Fatty acid oxidation is decreased, so you have a decrease in basal oxygen consumption rate, maximal oxygen consumption rate, ATP coupled respiration, but you have no change in non-mitochondrial respiration. So what this tells you is that for astrocytes to ramp up all of these normal metabolic processes when challenged, they have to have the ability to transport citrate between the cytosol and mitochondria effectively. So the last few experiments I want to show you are the Transwell system I told you about. So what we did is we took neurons and plated them in a dish, and we treated them with Tat, NMDA, alpha tocopherol, the normal stuff. We collected the cell-free media as we have been doing. Then we took a Transwell system with astrocytes on the filter and different neurons, fresh neurons, in the dish below. We put this neuron conditioned media on the astrocytes so they could start processing all the fatty acids that came from these neurons. Now we wanted to see when the astrocytes are busy processing all of the fatty acids from the challenge neurons, what happens to healthy bystander neurons, if there are any. So in this case, what we did is we collected these neurons and looked at them for fitness. Okay, were they alive? Did they have normal levels of MAP2, of PSD95, etc.? And in some of these astrocytes, we also knocked down the citrate transporter. So the reason I wanted to show you this is because I get very confused when I try to explain what we did. And I just want you to see, where we're going with this. So what we did then is, remember, we had our neurons, astrocytes, neurons, and we're now assessing the neurons, not the astrocytes. Okay, so if we take the neurons and harvest them and run Western blots, what we see, first of all, and this is important, is regardless of whether or not the citrate transporter was knocked out in our astrocytes in the Transwell, it has no impact really on the levels of this transporter in neurons, which it shouldn't if everything worked properly. We do see that PSD95, which is a marker, a synaptodendritic marker in neurons, goes down if the astrocytes weren't able to process the citrate and feed the neurons below what they need to. We see that MAP2 goes down, and we see that ATP goes down from neurons that were being taken care of by astrocytes that couldn't transport citrate. We also saw that neurons, some of them died. So this points out to us that astrocytes, not only do they shut down some of their neurotrophic activities, they also need to be able to transport citrate to do anything at all. So I know I've thrown a lot of data at you, and this is where we are right now with our project. And like I said, with Dr. Khalili and Dr. Howie, we are expanding this study using eco-HIV mice. Norm and I just got a R56 that'll be an R01, and this is what we're doing in this grant. And all the data I showed you was used to convince NIH and the reviewers that it was actually worthwhile. And I'm really excited because this is so much fun. So what I've told you is that when you challenge neurons with coke and tat, mitochondrial reactive oxygen species goes up, the neurons induce autophagy, and they produce and expel excess peroxidated fatty acids. When astrocytes are challenged with coke and HIV directly, they increase their metabolism. They switch to beta oxidation for their energy source. They decrease cholesterol, lactate, ApoE, and ABC1. Essentially, they start taking care of themselves, and it becomes all about them now. They don't really care about the neurons, and any little bit of lactate or cholesterol made is fine. But what we've shown is that when you knock out the citrate transporter, it gets even worse. I just said that here. Not only does it get worse for the astrocytes, they can't process what they need to, and they start accumulating these toxic lipid droplets that contain peroxidated fatty acids, but the neurons, the bystander neurons, are also suffering from this. Decrease neuronal fitness, etc. So that's all I have for today, but it's a really cool story that I enjoyed telling you. I want to make sure that I credit the people who actually did the work. Dr. Srinivasan and Dr. Kodo did most of the groundwork for all of this. Dr. Garcia, he helped out with his new data. We have Fergan and Caitlin, Ph.D. candidates in the lab that are there helping too. An undergrad, Ariba Rahman, and of course, Norm Howie, David Volsky, Kamel Khalili et al. And our funding down here, I think you can probably see it, a T32 that Dr. Berto is running for us here for these wonderful students that we have. Thank you so much for your time, and I will take questions.
Thank you, Diane. Absolutely wonderful talk. We have two questions in the chat. One is for me, so we can skip over that and give others the opportunity. First is from Corey White. Corey, do you want to unmute yourself and ask the question directly?
Yeah, I can. So I was just curious if you've observed any changes in ACOT7 expression in neurons due to either like cocaine or tat.
I haven't looked at it. Corey, could you tell me something about the ACOT7, please? I'm not familiar with it.
So it's a acyl-CoA thioesterase that is specific to neurons, and it's been shown to be necessary for like cellular retention of like acyl-CoAs themselves. So for like retention of like the fatty acids. So I was curious if it was like necessary for the expulsion, like to have like an increased regulation of like the peroxidated fatty acids.
I do not know, but I think that's an excellent idea. Where are you from, Corey?
I'm at Johns Hopkins in Dianne Williams' lab. Yeah.
Hi. Yeah, I would love to look at that. And since you're at Hopkins, you know, we definitely, well, I mean, we could collaborate anyway, but if you would like to look at some of this, we would be happy to work with you to do that. That's a great idea.
Yeah, thank you, of course.
Let's see, I'm just going to skip over me. Jonathan Geiger, if you would like to unmute yourself, please ask your question.
Sure, Diane, hi.
Hey, Jonathan.
So there are different populations of mitochondria, and the perinuclear mitochondria are really thought to be especially metabolically active, and not just for metabolism, but also for protein translation.
Yes.
Have you looked at sort of different locations and populations of mitochondria in your responses?
Not in this study, but Dr. Srinivasan, who I think is on the call right now, he has looked at different populations of mitochondria in the context of cocaine, and he can probably answer some of that. In these studies, I have not. Dr. Srinivasan, are you on? Okay.
Hi. So we looked into the mitochondria, the different populations in another study. So what we did, we find when we treat the astrocytes with Tat or with cocaine or Tat plus cocaine, so we found the mitochondrial shape changes, and we also found perinuclear appearance of the mitochondria. And so that we really looked into that, and we really found that that's a calcium mediated and a DRP1 mediated mitochondrial fission is undergoing when we treat the astrocytes with cocaine or Tat plus cocaine. So that is a different study. So there we generated a lot of data in some other contexts. I think either me or Dianne can talk about that.
And if I may, can I ask one follow-up question? And that is that, I mean, I have nothing against mitochondria. I like them. They're important, but they don't work in a vacuum in a cell. And modern cell biology is very focused these days on interorganellar signaling, and both physical and metabolic. So I'm just curious whether you did anything with other organelles simultaneously with the mitochondrial work.
Yeah.
Actually, I was thinking I was asking Dianne, but
Oh, sorry, I did something weird.
Well. Either one.
Srinivasan, you can answer, or
Yeah, we are also looking into the contribution of ER along with the mitochondria when we study the mitochondrial derived vesicles. So the signal is coming from the ER through IP3 receptors, etc. So that also we are really looking into that. And due to that signal, we found that apart from lipid and other paracrine factors, there is a lot of microRNAs also getting released through that vesicles. So that's passing a lot of signals to the neurons and some other brain cells also.
Okay. Both of you for answering the questions.
Thanks, Jonathan. Good to see you.
Yeah, you too.
Thanks, Jonathan. The next person is listed as Heather Thomas. I'm pretty sure it's not, though. Question begins with great talk. So whoever that is, could you please unmute yourself and ask your question?
Yeah, I think so. I'm Pragnay. Hi, Dr. Diana. Great talk. So my question is, so have you seen any changes in glucose metabolism in this condition with cocaine and Tat?
Yes. So what we see, I didn't present it to you today, but when you take astrocytes and treat them with cocaine or Tat or both together, and this was, don't remember if it was Dr. Srinivasan or Dr. Koto's paper, it was one of them. It showed that, so if you're looking at glucose metabolism, what happens is it comes through, and then pyruvate, instead of going through LDH and lactate, it kind of switches over to go straight to acetyl-CoA into the mitochondria and switches from the normal pathway of glucose metabolism to beta oxidation. So that's what we found. And like I said, that's published, and I think back in some of the other slides, I'll show you right here. Right down here at the bottom, if you look at Koto and Glia and Nataraja Srinivasan in Cell Disease and Cell Death and Disease, and I noticed I spelled his name wrong. It's supposed to be an N. You can definitely find figures that support that.
Okay, but there will be huge ATP production then, because
Yep.
Fatty acid also driving for oxidation to produce ATP and
Yes.
glucose.
Yes.
Okay.
Absolutely. They make a lot more ATP.
Okay.
So close to
Okay.
Okay, thank you.
Thank you, Pragni. And the next question in the chat is from Mutha Kumar. I think hope I'm pronouncing that correct.
Yep. Hi, this is Muthu from Dr. Buch Lab, UNMC. Great talk. So I have a question. So how the intracellular levels of cholesterol are reduced by Tat or cocaine? So what would be the possible reason for this?
So thank you for that question. So if you look at this slide right here, this is Bianca Cotto's paper. So what she discovered is that what happens is when you treat with cocaine and Tat, you have glucose coming in, pyruvate gets shuttled to acetyl-CoA, you have disruptions in HMG-CoA pathway here that decreases cholesterol. This is also linked to LXR, nuclear receptor mediated transcription. So it feeds back in this mechanism here. This pathway, HMG-CoA, this pathway is what is dysregulated. And in Bianca's paper, you can see figures that break down this pathway more specifically. And hers is in GLIA in 2018.
Okay. So it says it has a defect in the synthesis. Okay, thank you. So, okay, one more question. Okay, so you showed the HIV Tat is also increases the levels of ATP, the production of ATP. So do you think it upregulates the functions of mitochondria? So what do you think about the Tat is being
I'm so sorry, I didn't understand that
No, the
question.
HIV Tat, it increases the mitochondrial respiration, right? Increases the
Yes.
yeah. So what do you think? It's Tat basically upregulate the mitochondrial function?
Norm, can you help me out? I can't understand the last bit of the question.
If I understood myself correctly, he's asking if Tat itself enhanced mitochondrial function. Is that correct?
Yes. Yeah, that's the one. Yeah.
Norm, I'm having trouble hearing. Say that one more time.
He's asking if Tat itself enhanced mitochondrial function, increased mitochondrial function.
Yes. Not to the levels that cocaine did, but yes, it did.
Okay, thank you.
All right, thank you. Ron, you're up.
Sorry, I always enjoy listening to you, Dianne. Just so easy talking and very, very clear for somebody who's not a basic scientist. But I was wondering if you see at this point if there are any clinical implications. The one that pops into mind, of course, is in people vitamin E, which hasn't worked in a whole bunch of other conditions. And as you know, people are spending billions of dollars on vitamin supplements that probably aren't helping them at all.
Yes.
Or is it just way too early to go there yet?
I don't think it's way too early. And the reason I say that is back when Bianca was in the laboratory and she finished in late 2017, when she was looking at the LXR nuclear receptor signaling over in the pathway that we were just discussing, we did use vitamin E to disrupt some of the dysregulation that we saw, and we were going in the direction that to expand that because it did seem to help. It was it wasn't just straight vitamin E, it was some breakdown product or so. I don't remember exactly what it was called, but I don't think it's too early, and I think it might help. We would just need to revisit that. Bianca graduated, so we kind of went a different route.
Well, you're working with David Volsky, so you have access to more than just a Tat exposure model, and you can take those animals and see if you can make them better with some vitamin E or some other.
That's a really good suggestion. I can definitely do that because we have the eco-HIV mice going here with and without cocaine, but I'm interested to see what would happen with just HIV in the context of abnormal aging in HIV, in people with HIV, and the exacerbated accumulation of lipid droplets in glial cells, even in normal aging. So that's a very good idea. Yes, I have to write that down.
Thank you, Ron and Diane. Kelly Jordan Shudo.
Hey, Diane, great
Hi,
to see you
Kelly.
and great to hear
Hi.
about your research. I know it's been too long.
Yeah.
Really exciting stuff. And so my question for you, which means that I also want to talk to you offline about this as well, is what do you anticipate the effects of this change in astrocytes would have on other cells like microglia and oligodendrocytes? And of course, that's a loaded question because we know how important astrocyte production of lipids is for oligodendrocyte development and function. So we've been thinking a lot about this in some of the work we've been doing. And so I'd really love to hear your thoughts on that.
So microglia, I know that first of all, we haven't looked at either of those cell types, but I know microglia are another culprit in the accumulation of lipid droplets with normal aging and can become problematic in disease impacted, like age-related CNS diseases. So microglia are a culprit. Now, oligodendrocytes, I read, nothing's been done in the context of HIV with oligos, but in other diseases, only in complicated mouse models, have a few people said oligos are impacted by this really bad and it's not a good thing. So it's wide open, Kelly.
Thanks, Diane.
Thank you, Kelly. If there are any more questions, please just unmute yourself and ask the question. Otherwise, we'll close in a minute. Okay, then we will just take the last moment to thank again Diane for a very interesting, highly stimulating talk. Very much appreciated. We are on again next week, same time. The speaker or speakers will be announced hopefully later on this week. So thanks, everyone. Stay safe. We'll see you in a week.
Thank you. Bye. Good to see everybody. Bye.